the remaining part of the second year portion, that is aquaculture, poultry and miscellaneous topics. What are the miscellaneous topics? One is the introductory part of biotechnology, remaining part of biotechnology. After biotechnology, we have to discuss the cell cycle and its regulation, stem cells, and next the last one is biomedical technology. Again repeating, from today onwards we are going to discuss the miscellaneous parts of the second year syllabus. One among them is aquaculture. From the aquaculture we are getting one question. From the poultry we are getting one question, and the remaining part of biotechnology, that means introductory part of biotechnology, remaining part of biotechnology. After completing the biotechnology, the cancer biology, cell cycle and its regulation, the stem cells, all these are the miscellaneous topics. From these topics we are getting one question. And the last, biomedical technology, X-rays, CAT, scan, and next HIV test, and next ECG. From this topic, that is biomedical technology, we are getting one question. And all these topics are the generalized topics. These topics are not generally the much more, these are not the generally much more concept oriented, a general discussing part. Now the first one is aquaculture, a small introduction regarding the aquaculture. What is meant by aquaculture? The rearing of any type of aquatic organism. Generally, while we are discussing the aquaculture, we have to use the blue revolution. Now what is the blue revolution? The method of capturing and culturing of aquatic organisms, that we call it as blue revolution. In the aquaculture, one part is the fin fishery, the rearing of aquatic organisms having fins, called fin fishery. And the next one we call it as shell fishery, the rearing of aquatic organisms having the shells, that we call it as shell fishery. One example for the shell fishery, that is lobsters, crabs, prawns, molluscan animals. In the first what we discussed, fin fishery, the rearing of aquatic organisms having fins, that we call it as fin fishery. That means the rearing of variety of fishes. Now we have to discuss some very important bits regarding aquaculture with reference to EAMCET point of view. Now as we observe the first one, essential amino acids that are abundant in fish meat. From the olden days, the fish meat is considered as the brain food, because every day if we are going to take a small part of fish body, that means 100 grams of fish flesh is taken, that fish meat activates our brain cells. In the central nervous system, the neurons are present, we know very well. Every day if we are going to take 100 grams of fish meat, such food material activates the neurons of our central nervous system. So the brain cells are working at sharp. So the fish meat is popularly known as the brain food, because What are the essential amino acids that are abundant in the fish meat? The best is the methionine and lysine. They are the amino acids. Students, here generally we are doing a mistake. Here what we are asking, the amino acids. We are having an idea regarding the genetic code. The essential amino acids and non-essential amino acids. Essential amino acids play an important role in our day-to-day activities. Each amino acid is generally recognized by one triplet codon. So that while preparing this question, what are the amino acids present in the fish meat? Methionine, lysine. Better you have to refer the genetic code regarding the part of genetics. In the genetic code part, we are having the triplet codons for different amino acids. So regarding the methionine and lysine, you have to check what is the triplet codon which are present in the genetic code. And now the answer is the methionine and lysine. Now the next one is Chutikaran, coast of Tamil Nadu, is famous for. Now as we observe the aquaculture, one we call it as the freshwater aquaculture, the marine water aquaculture, the brackish water aquaculture, and seaweed culture and pearl culture. And you know very well pearl culture, that means the preparation of pearls, muchamulu. Now as we observe the pearl culture, regarding the pearl culture in India, particularly with reference to Tamil Nadu, in the Tamil Nadu area near Chutikaran coast, there we have to observe more and more pearl culture industries. In the world, what is the first place goes to the pearl culture? That is the India. And the second place goes to the Japan. Generally for the culture of pearls, we have to use Pinctada vulgaris and Pinctada fucata. These two are the marine organisms. Recently, another organism is also used for the preparation of pearls, that is Lamellidens marginalis. That is a freshwater form. Again repeating, one we call it as Pinctada vulgaris, second one we call it as Pinctada fucata. These two are the marine organisms. The third one we call it as Lamellidens marginalis. The culture of Lamellidens marginalis has been conducting by Central Inland Freshwater Aquaculture, which is at Chennai. These three organisms are used for the pearl culture. Generally, as we observe the pearl culture, if any foreign particle enters into the body of Pinctada vulgaris or Pinctada fucata or Lamellidens marginalis, generally the foreign particle does not enter into the body, because in these animals' body Aspergillium is present. The Aspergillium tests the purity of water. So the Aspergillium does not allow any foreign particle enters into the body. But here it is a culture. In the culture, what is going on? As we observe the Pinctada vulgaris, Pinctada fucata, Lamellidens marginalis, their body is covered and protected by two shells. these two shells are attached by a hinge joint. These two shells are not opened, but in the culture process, the workers open the two shells. With the help of forceps, they introduce one sand particle or any particle. Now the two shells are closed. After few days, what is going to happen? In these animals' body, the mantle is present. We know very well it is a layer. The mantle secretes a substance around the foreign particle. Because of this secretion, the foreign particle becomes pearl. So that is the process going on conducted in the pearl culture industries. Now, Chutikarna coast of Tamil Nadu is famous for, you have to observe the answer, that is the pearl culture. Now we go for the next question. List 1. Now for the EAMCET point of view, as we observe the previous papers, regarding the aquaculture in the previous EAMCET examinations, we are getting questions from the fish, gear and craft. What is meant by craft? Different vessels, boats are used to transport the aquatic organisms as well as to capture the aquatic organisms. One is the Kakinada Nava, Masula boat, dugout canoe, Dhoni. All these are the examples of craft. You prepare this paragraph more and more times, because from the craft and gear we are getting the questions. What is meant by gear? Different nets. In Telugu they are called valalu. Different nets are used to capture the aquatic organisms. Some nets are used by one fisherman. Some nets are used by two fishermen. Some nets are used by more than one person. And some nets are operated by mechanized boats. Now you observe the list 1. Long line net, boat seine, gill net, cast net and stake net. Generally long line, such net excellently operated by USA fishermen. In the remaining countries, the fishermen are unable to operate the long line net, but the USA fishermen can operate excellently. The long line net is used to capture the tuna fish. If we go for the USA, in the United States of America, as we visit any restaurant, there we are getting the tuna fishes. In the tuna fishes meat, the omega-3 fatty acids are present. The omega-3 fatty acids, they dissolve the cholesterol. They are very popular at USA places. Nowadays, the tuna culture has been popularizing in remaining parts of the countries. And long line net is used to capture the tuna fishes. The second one is boat seine. It is used to capture inshore fishes. What is meant by inshore? The method of capturing fishes very close to coast or very close to kinare. You know the meaning of the coast? It is also called the kinare. Near the coast, if we are capturing the fishes, we have to use the boat seine. What about the gill net? It is a wall-like net. It is operated by two persons. The gill net, it is used to capture the oil sardine, that is a fish. Its scientific name is Sardinella longiceps. It is popular in the Gujarat state coastal area. The byproducts obtained from the oil sardine or the Sardinella longiceps are used in the preparation of so many food materials. In the same way, they are used in the preparation of different soaps, paints, etc. It is a repeated question in the main EAMCET examination. The gill net is especially used to capture the sardines. What about the cast nest? It is used to capture the fishes in the freshwater. The freshwater bodies also called inland water bodies. In order to capture the fishes in the freshwater bodies, we have to use the cast net. Generally, it is operated by one fisherman. And the last one is the stake net. It is used to capture the fishes in the brackish water. In Telugu, it is called Uppu Neeti Kayya, or the water present at river mouth. In Telugu, it is called Nadi Mukhadwaram. The place where the river enters into the sea, that we call it as the river mouth. In the river mouth, two kinds of waters are present. One is marine water and one is fresh water. That means at river mouth, these two waters are mixed. That is called the brackish water or eucerine water. There, in order to capture the aquatic organisms, we have to use the stake net. Now the answer is, you observe. Now we go for the next question. Culture of murales is discontinued. As we observe the murales, the Chana species, nowadays their culture is being stopped because the place where these murales are cultured, in those places the water is poisoned. In those places the remaining aquatic organisms are going to die. That means the place where murales are cultured, in those places if other aquatic organisms are present, because of the murales the water is polluted. In that polluted water the remaining aquatic organisms are going to die. So nowadays the culture of murales, a type of fishes, is stopped because they have been suffering from ulcer in the alimentary canal. That is technically called epizoottic ulcerative syndrome. What is meant by syndrome? A group of symptoms. Epizoottic ulcerative. In these fishes in the alimentary canal, the formation of ulcers. Now you observe the assertion. Culture of murales is discontinued because heavy losses are incurred by epizoottic ulcerative syndrome in these fishes. Now the answer is, both are correct or explain CA. Now the next question. Read the following and choose the correct one. Now as we observe the development of fishes, in the development of the fishes, the sperm and ovum will undergo fertilization. We are getting the egg. In the presence of sunlight, the eggs are hatched out into larvae. Depending upon the size, they are named as fry, fingerling, etc. In the development of fishes, we have to use different ponds. One is the nursery pond, the rearing pond, the stocking pond. The stocking pond is classified into pre-stocking pond and post-stocking pond. Now you observe. What is the first column, second column and the third column? Fry. What is the size of this larva? 2.5 centimeters. Where it is? It is growing in the rearing pond. What about the fingerling? Okay, the size of fingerling is 4.5 centimeters. But where it is? Nursery pond. No. The first pond is nursery pond. In the nursery pond, the larvae are very, very minute. For how many days they are in the nursery pond? Just hardly for 15 days. The nursery pond is followed by rearing pond. The fry and fingerling both are present in the rearing pond. But here, the fingerling present in the nursery pond. That is the wrong. Third one, ulva. That is one type of seaweed that is named as the green algae. How it is developed? By a process called seaweed culture. We know very well the red algae, the brown algae, the vulva and spirulina. Now the vulva, it is a type of seaweed popularly known as the green algae. How it is developed? By a process called seaweed culture. It is a seaweed. In Telugu it is called kalupu mokka. And the fourth one, sargassum. The second column is red alga and seaweed culture. But the sargassum, it is not the red algae, it is the brown algae. So based on this information, you check the answer. Fry, the size is 2.5 centimeters. It is growing in the rearing pond. And vulva, it is a type of seaweed popularly known as green algae, and how it is developed by a process called seaweed culture. Now the next question. Following are the various steps in the fish culture. Arrange them in a sequence. Now, we are going to culture the aquatic organisms. How we have to culture the fishes? First of all, we have to select the brood fishes, the male fish and the female fish. Generally, what is the breeding season? That is rainy season. But we are having the marketing in all seasons. Other than rainy season, if we are having the marketing, how to get the product from these fishes? First of all, we have to select the male and the female fishes. The fishes that are selected by the culture are called brood fishes. Now, we go for the hypophysation technique. As we observe the fish markets, particularly in the Visakhapatnam area, as we observe the fish market, there we have to observe some fishes without heads. That means the workers remove the head of the fishes. The head is removed. Now they break the skull. That means in the skull one part is cranium. The cranium is removed. As we observe the brain on the ventral side of diencephalon, what is the present? Pituitary gland. Now the workers take pituitary gland in a plate. They add few drops of alcohol. In the presence of alcohol, the pituitary gland is crushed. Now the entire solution is taken in a test tube. Now the test tube is kept in a centrifugation machine. Now the machine is on. Centrifugation is going on. After completing the centrifugation, we are getting the extract. Such extract consisting the gonadotropic hormones. Now we inject the gonadotropic hormones into the body of male fish and female fish. In the presence of hormones in their body, the testes and ovaries, they germinate or they are activated. They release more and more sperms, more and more ova. Now they undergo fertilization. We are getting the zygote. In the presence of sunlight, they are hatched out into larvae. Now the larvae first of all kept in the nursery pond. Then they are shifted to rearing pond. Then they are shifted to stocking pond until they are marketable. That is the process what we are going to do in the culture of the fishes. Now, Following are the various steps in the fish culture. Arrange them in a sequence. First, you observe the statements: stocking pond, rearing pond, collection of brood fish, hypophyization, and nursery ponds. Now the first one is collection of brood fish. Then we go for the hypophyization technique. Then the larvae first kept in nursery pond. Then they are shifted to rearing pond. Finally, they are shifted to the stocking pond. Now, what is the answer? The answer is the third option. Now, the next question. Regarding the diseases of the poultry. It is very important regarding the main EAMCET examination. As we observe the previous EAMCET examination papers, particularly regarding the poultry, from the diseases part, we are getting the questions. The diseases are caused by protozoan parasites, helminth parasites, and the diseases caused by bacteria, the diseases caused by viruses, the diseases caused by the fungus. What are the bacterial diseases, viral diseases, and fungal diseases? Just you have to listen. Ranicate or Newcastle's disease. The second one we call it as Marek's disease. The third one we call it as Gumboro disease. And the next one is fowl cholera. And the next one is the coryza or row. And the next one is chronic respiratory disease, aspergillosis, aphilotoxicosis, and thrush or moniliasis. Again I am repeating. Ranicate or Newcastle's disease, Marek's disease, Gumboro disease. Gumboro disease is repeated main EAMCET. And the next three diseases, the fowl cholera, coryza or row disease, chronic respiratory disease. Fowl cholera repeated in the main EAMCET examination. The last three are the fungal diseases. One is aspergillosis, and the second one is aphilotoxicosis. The third one is thrush or moniliasis. Because of these diseases, we are going to get heavy losses. Now you have to observe name of the disease, caused by the organism, and what are the symptoms. Now you observe ranicate. It is caused by paromyxavirus. What are the symptoms? Respiratory and nervous disorders. Because of this disease, more and more young ones are going to die. We are going to get heavy losses. The mortality, the death rate is more. Now the first one is correct, because ranicate or Newcastle's disease caused by paromyxa, and the death rate is more. What are the symptoms? Respiratory troubles, nervous troubles. Second, aphilotoxicosis. It is caused by Aspergillus flavus but not fumigatus. So the second one is wrong. Third, fowl cholera. It is a bacterial disease caused by Passerella avicida. What are the symptoms? As we observe the cock or hen above the head, what is present? That means a cap-like structure. That means comb. And below the neck, what is present? The wattles are present. And now the third one is fowl cholera. It is caused by a bacterium called Passerella avicida. What are the symptoms? The discoloration of comb. What is meant by comb? It is present above the head. Discoloration of combs. So the third one is correct. What about the thrush? Thrush is caused by Aedeum albicans but not IBD. So here, what are the correct statements? Ranicate and fowl cholera. Now the next question. A poultry bird which is richly, you have to observe RICHLY. A poultry bird which is richly supplied by fat diet with less fiber will face the following problem. While we are giving the food material to the poultry birds, along with the food materials we have to supply the fibers food. In the food material, if more and more fibers are present at the time of laying eggs, it does not get any trouble. But here, in order to get benefits, the entrepreneur, that means a breeder or a businessman, he is giving the fatty material along with the food materials. So at the time of laying eggs, now this is the cloacal aperture. At the time of laying eggs, along with the egg, the oviduct and uterus will come out and they do not enter into the body. At the time of laying eggs, along with the egg, the oviduct and uterus come out. That we call it as the prolapse. In Telugu, it is called Meya Veyuta. That means at the time of laying eggs, along with the egg, the oviduct and uterus come out. They are unable to enter into the body. Such type of body is not eaten. Now here what is the problem? Along with the poultry feed, while we are giving the food material to the poultry birds, in the food material more and more fatty material is present instead of the fibers. So here a poultry bird which is richly supplied by fat diet with less fibers will face what type of the following problem, that is the prolapse. Now the next question. Broiler hybrids are produced by crossing. We are having two types of the birds. One we call it as layer and the second one we call it as broiler. What is meant by layer? Generally it is meant for laying the eggs. What about the broiler? It is generally meant for the meat purpose. In the market, we are going to observe hybrid broiler, hybrid layer. Here what is the question? Hybrid broiler is obtained by crossing the female Plymouth Rock or female New Hampshire to that of the male Cornish. Now generally while discussing the birds, for the female bird we use the word dam. It is not an Nagarjuna dam, etc. dam. While explaining the female birds we have to use dam. While explaining the male bird we are going to use the word sire. Now here the question is, how we are getting the hybrid broiler? By crossing the female Plymouth Rock or female New Hampshire to that of the male Cornish. Here Plymouth Rock and New Hampshire come under the American class. They are present in the American countries. And Cornish, it is present in the English countries, that is Britain countries. Now here the answer is the fourth one, dam, that is the female Plymouth Rock or female New Hampshire to that of the male Cornish. Automatically we are getting the hybrid broiler. Now the next one. Arrange the following mash. You have to observe the question. A small mistake. Mash. Arrange the following mashes. Mashes means the food material. Now as we observe in our houses, the mother is going to prepare some type of curries. For example, while preparing wankai curry, in the wankai curry our mother has to use batanis. So wankai curry plus batani. Generally we remove the batanis and take. But here what is the mash? While preparing such type of curry, the mother makes the wankai and batani in the form of a paste, that we call it the mash. In the same way, in the poultry, the entrepreneur or the owner supply the ingredients to the poultry birds in the form of mash. as we supply the ingredients in the form of the mash, they take all ingredients and grow properly. In order to get a better growth, the owner supply the ingredients in the form of mash. Now, you observe the question, arrange the following mashes in an ascending order based on the energy values. Now, as we observe, the first one is layer mash. Because of the layer mash, we are getting how much percentage of energy? 2500 kilocalories. Now, finish mash, it is giving to the broilers. Because of the finish mash, we are getting 3400 kilocalories of energy. Starter mash, that is also supplied to the broiler birds, we are getting 3200 kilocalories of energy. And chick mash, from the chick mash we are getting 3000 kilocalories of energy. And grower mash, we are getting 2700 kilocalories of energy. Now, based on the energy values, you have to arrange the food materials in an ascending order. That means bottom to top. Now, the first one is 2500 kilocalories. Now, this is the layer mash. Then we go for the 2700 kilocalories. That is the grower mash. And 3000 kilocalories of energy. That is the chick mash. And the starter mash, it is given to the broilers, 3200 kilocalories. This is the starter mash. The last one is finish mash. From this we are getting 3400 kilocalories. Now, based on this information, you have to observe the correct answer for this question. That is the fourth option. So, based on the energy values, we have to use the food materials in a ascending order. Now, the next question. Disease of poultry animals caused by protozoan called Eimeria tenella. This is the protozoan parasite come under the class Telosporia. Regarding the protozoa, subphylum Sporozoa, one class is Telosporia. Under class Telosporia, one example is Eimeria tenella. It is a protozoan parasite. Generally, this parasite attacks the poultry birds in the early stages. That means in the first week, the birds are going to get, or the birds are going to suffer from this parasite. This parasite causes coccidiosis. What is the symptom of the coccidiosis? Blood motions. Along with the fecal matter, more and more amount of the blood will come out, and the mortality rate is very high. More and more birds are going to die. Now, the disease, just you have to check the word, disease of poultry animals caused by protozoan called Eimeria tenella is, such disease is the coccidiosis. The symptom is blood motions. Now, the next one. The controlled use of biological agents or certain cellular components for the beneficial purpose is activity under. Now, we are discussing the biotechnology. What is meant by biotechnology? In Telugu, it is called Jiva Sanketika Shastram. Biotechnology, the technology associated with the biology. Now, what is meant by biotechnology? We have to use the microorganisms for the welfare of mankind, or we have to use the cellular components for the welfare of mankind. Because of the applications of microorganisms, we have to prepare the alcohols, we prepare beverages, we prepare different organic acids, citric acid, lactic acid, glutamic acid. We have to prepare the beer, the wine, the whisk, the rum, the gin, and we have to prepare the alcohol, and we have to prepare the gasohol. What is meant by gasohol? It is the mixture of petrol and alcohol. It is used as a fuel in the Brazil country, the most popular fuel. By using the microorganisms, we have to prepare the vitamins, we have to prepare antibodies, antibiotics, monoclonal antibodies, different vaccines, and different hormones. All these are prepared by using the microorganisms or cellular components. That we call it as the biotechnology. Now the answer is biotechnology. What about the biochemistry? The chemistry related to the biology. Because of the biochemistry, we know what is meant by osmosis. We know what is meant by diffusion. We know what is meant by membrane transport. We learned in the first year, first unit. Relation between the zoology and other sciences. What about the bioinformatics? By using computer knowledge, we have to analyze the botany information, the zoology information. And what about the bioenergetics? The bioenergetics explain how the living organisms get energy, how they store energy, how they utilize the energy. Now the next question. The industrial use of microorganisms. In the biotechnology, we have to use the microorganisms in the industries. What are the microorganisms? Different bacteria, different viruses. Here the students may get a doubt. Why don't we use the human beings in the biotechnology? The human being is the well-developed animal. Suppose we are conducting experiment on the body of the human being. If the experiments are failed, what is going to happen? The human being is going to die. Once the human being is going to die, who is responsible for the death of human being? So the well-developed animals are not used. So the microorganisms, they are small in size. They are called the miniatures. The microorganisms are easily adaptable to the industrial environmental conditions. Again repeating, the microorganisms are easily adapted to the industrial environmental conditions. So we are going to use the microorganisms. So here the industrial use of microorganisms is one aspect of. Now you imagine the answer is biotechnology. Now the next question. Now in this question, one more is, what is meant by biometry? In order to explain the biological information, we apply the mathematics. Application of mathematics, that we call it as biometry. Now the next question. The technique for cloning genetically engineered molecules in foreign cells. Now you have to observe the question. One is foreign cell and the second one is genetically engineered. That means here we have to use the recombinant DNA technology. The technique for cloning genetically engineered molecules. That means the molecules prepared by recombinant DNA technology, they are used in the foreign cells. Such technique is developed by Boyer and Cohen. Generally the students are doing the mistake here because where these two words are present. As we refer the biotechnology topic in the second year. In the second year biotechnology topic in the first page, left side, that means top of the page toward the left side, these two scientist names were present, Boyer and Cohen. They developed this technique. Now the answer is the Boyer and Cohen. Now the third one is Jacob and Monod. Already we discussed in the genetics. The gene expression and regulation in the prokaryotes, that is operon concept proposed by Jacob and Monod. Now the next question. Recombinant DNA technology. that manipulates the genes or genetic nature of organisms is commonly referred to as. Now as we observe the recombinant DNA technology, what is meant by recombinant DNA? Now this is one kind of organism and this is another kind of organism. You have to follow the concept. This is one kind of organism, this is another kind of organism. Now from this organism we have to take one strand of DNA, a part of DNA. From this organism we have to take one part of DNA. For your kind information, to identify the difference, I used these. Now this is one fragment of DNA derived from one source, and this is another fragment of DNA derived from another source. This is one source, this is another source. Now these two are fused. Now we are getting this result is named as the recombinant DNA. The word recombinant DNA is applicable to the result obtained from the two DNA strands derived from two different sources. Now, recombinant DNA technology that manipulate the genes or genetic nature of organisms is commonly referred to as. Now the answer is the genetic engineering.